All right. Hello. Hello. Okay, good. All right. So this is our last portion. And this is about the home hemostasis. So we talked about this terminology already. So this is about to stop bleeding. This is important because once in a while we will get injured, and we need to be able to stop bleeding in order to we're gonna have a leakage, right? So we don't want to have a leakage. So we have a system to stop bleeding. Now, what we are going to talk about is what is this system? Hemostasis. All right. This system has three major players. One is the vessel. So the injured vessel need to send out signal in order to trigger that hemostasis process. Second is the platelet. So platelet's major function is hemostasis. And the third one is fibrinogen. So we talked about these three already. In the earlier lecture, we talked about the cardiovascular system. So we know vessel. We know vessel has three layers: tunica intima, tunica media, tunica externa. And so when the endothelium cell is injured, then the blood will see something that they are not supposed to see, right? Because everything should be covered by endothelium. Once they see it, the vessel will send out that using it as an opportunity to send out signal. And then vessel will also do the constriction in order to reduce the blood to go through it. And then the vessel will also send out signal to activate the platelets. It will go through several other pathways to finally activate the fibrinogen. Platelet is a blood cell, a type of blood cells. Fibrinogen is a type of plasma protein. So we talk about these in the earlier portion of this lecture. So this is kind of put things all together and then to talk about these process, hemostasis. So that's that. All right. So let's get started. So hemostasis has three major players: the vessels, the platelets, and fibrinogen. And it also each one will also has its contribution to hemostasis. So the vessel by itself, you can see that here is the tunica intima, tunica media, tunica externa. So here we have the smooth muscle right here. So muscle constriction. So what does vessel do? It does vessel constriction. It causes the vessel constriction. So vessel constrict and also reduce the blood flow, which is needed. The second one is the platelet activation. So platelet right here. So you can see that these platelets are smooth. Yeah, so their surface is smooth. And so they freely diffuse. They don't get aggregated that easily. Otherwise, we will form the thrombosis, which is not good, right? So we will not bother them, and they will not be bothered. So they freely flow freely throughout our circulation. But when they see something in the vessel, usually it's collagen. We don't see collagen in the blood. So collagen is one of the trigger. And when collagen is exposed to these pellets, it will poke it. When pellet flow through, see the collagen, pellet will get exposed, will get activate. So and so these pellets will become, when they are activated, they will do two things. They will become sticky. So they used to be very smooth. They don't interact with each other, but now they will expose something on the surface. So they will get easily to get interact with each other. They basically bond with each other. And then another thing is that that's very second thing is that these pellets will also release some other chemicals, they will spread that chemicals in that environment. So that chemical will serve the function to activate any platelets that happens to flow through. So even though they don't get interact with collagen, but since one of that is activated, they will release that chemical. So when one goes by, it will be activated. So they got stuck together. So that's that. So that's a pellet. Another one is fibrinogen. We talked about the fibrinogen in the earlier. Let me show you here. So this is the slides. So here is the blood in the very beginning. So plasma fluid right here. So plasma, you have albumin, globulin, and the fibrinogen. We also kind of saying it here, right here. Yes, plasma protein. This is one of the it's not the most abundant, but it's good amount. So fibrinogen is one. Its function is to stop bleeding, and fibrinogen, just like platelets, they don't easily get sticky. They don't attach to each other. And as you can see that this is something gene, so this is not active form of this protein. And so that's that. So fibrinogen will get activated. When they get activated, they will be converted into fibrin. So let me see that here. So fibrinogen, it will be converted to become fibrin, just like angiotensinogen to become angiotensin 1 and angiotensin 2. Fibrinogen will become fibrin, then fibrin will become sticky. So they will be, one will attach to the other one, and this since this is protein, so we call it is forming a polymer. So it's 1 unit, 2 units, 3 units, and connect together to become a polymer. Let me show you a picture. I think I have the picture like this one here. Yeah, like this one here. So here is a fibrinogen, and fibrinogen, once it's activated, then it will be some of those portions will be truncated. So these are just like a safe, once it's removed, it become activated, and then they will stuck together, and then you can see that it will become fibrin polymer to become fibrin cross-linked fibrin polymer. So that's fibrin. So once they stick together and they become the polymer, and it's like a mesh work. It's not just one dimensional, it becomes mesh work. So it kind of attached to the leakage site, just like a patch attached to it. So that's that. All right. So that's these three, and that's it. That's the major thing. You need to know what are the major player, which your followings are the major player for the hemostasis, vasoconstriction, platelet activation and aggregation, as well as the fibrin. Fibrinogen become fibrin become the fibrin network. Yeah, something like that. All right. So here, just put them all together, you can see that once there's an injury, then vasoconstriction becomes more. It doesn't really show that constriction that clear. This one shows the constriction very clear. And then you have the platelet coming here, fibrinogen become fibrin, and these red blood cell is because they just flow through, because here you have leakage, so they all flow through. Some of the blood flowing out, some of the blood got stuck between. And gradually, very quickly, you have a platelet activation, fibrin forming the coagulation, and so that's that. So first one is constriction. So you have the injured side. So here is the injured side. Then you have the constriction. You want to know, I also want you to know that any injury will cause the constriction. And that is why that hypertension, it will cause the injury. You can see that this endothelium cell is injured. They disappeared. Here you have the endothelium. It disappeared. So once this endothelium is is it doesn't form this inner layer. When it's broken, then anything will trigger the smooth muscle. It will cause vasoconstriction. Yeah, and which will lead to the hypertension, right? Constriction, TPR, total peripheral resistance increase the constriction, increase the blood pressure. Yeah, so that's that. And the next one will be the platelets. So you can probably see here. So here is platelet. The platelets are the fragments of a larger cell. This larger cell is called mega like large, karyocyte, megakaryocyte. So this megakaryocyte will trim off some of this portion, and this is the platelets. And so platelets is just like a fragment of these megakaryocyte. And in the same way, platelets don't have the nucleus. Two cells in the blood don't have the nucleus. Those are the red blood cell and the platelets. So and platelet is much smaller. You can see that it's only a fragment of these cells. So they are very small. Yeah. So about 200 billion platelets are produced daily. Each one live about 3 to 5 days. We don't really care about this recycling. We don't really talk too much about it. The amount, even though it's a lot, but compared to red blood cell, is much, much less. Besides that, the size is very small. It doesn't really cause too much burden compared to the red blood cell if we don't really take care of them very well. Yeah, so that's that. And you can see that it contains several components. Even though it doesn't have the nucleus, it still have the mitochondria. It has a lot of granule. So this granule basically is the pocket that store a lot of chemicals. And the two of the chemicals that we will talk about are the one that will influence the neighboring platelets in order to call them all together to form this clot. Yeah. So once it's activated, it will release it. So we call this platelet activation. How do we activate it? First, you need to have exposure of collagen. So collagen from the vessel wall, once it's exposed, so it can be your wound, it could be the internal, once you have the hypertension, glucose, virus, bacteria, etc., you cause the endothelium cell leakage. Once there's a leakage, the collagen inside of this vessel wall will get exposed, and that explosion will trigger platelets. Platelets can be activated by direct contacting the vessel wall components such as collagen. It can also be activated by the activated platelets. So once there is some platelets got activated, they will release something called ADP. This is adenosine diphosphate. So we talk about the ATP, right? Triphosphate. This is ADP. And so they will release it. And then it will also release another thing, which is thromboxane. So these two will get released. When they released, they kind of make that in the environment, and because the vessel is constricted, so it's not very huge blood flow to go through. So it's kind of like isolated area. It's not totally constricted, but it's smaller. So this chemical will be around, and they will affect the platelets when they go through. When platelets go through, they sense that ADP and the thromboxane, these platelets will become activated. They will get attached to it. These activated platelets will also release ADP and thromboxane, and that will cause these platelets become activated and also become sticky to each other. So I think I have a picture about it right here. So this is a platelet. So you can see that this is a platelet. These may be, in the beginning, they are not activated, but say this one is the first one, this is the first one, so this one coming here, attach the collagen in the vessel wall, collagen will trigger it. This is called von Willebrand factor. Don't need to worry about that. We are not going to talk too detail about this. This is just a mediator. There are many like cell adhesion molecule to get involved, but don't need to worry about that. You just need to know that the vessel will expose their collagen that will lead to the platelets get activated. Once they get activated, they become sticky. They will attach. They will bond to each other. They will also release thromboxane and ADP, and this one will affect any platelets which flow through. Even though these one may not directly touch the collagen, they will get activated by this ADP and the thromboxane. And so once they got activated, they become sticky. They will stick to all of them. So it forms like a clot, right? It will just get together and try to seal this damage. You can see that this doesn't really have the leakage, but any wound, any injury will cause them to get together to each other. So that's why that any vessel injury will increase the chance of forming this thrombosis. You need to know that once this one is formed, let me see this one here. Once this one is formed, all right, if we don't take care of it very well, the first is that we try to stop bleeding, but if we don't take care of this very well, they may flow away, and then they may get stuck somewhere in the other in other region. So it will be very dangerous that when this is kind of like a little like a rock, right? Thrombosis, then then they will flow. If you go to the brain get stuck in the brain, then you have stroke. If it gets stuck in the coronary artery, you have heart disease. So it will be kind of unpredictable. Yeah. So we need to take good care of that. Our body will take good care of that. All right. So that's that. All right, so that's the platelets. And the next thing will be the fibrin. I think so. Yes, fibrinogen. All right, so this one, we show you this one already. We need to activate the fibrinogen. Fibrinogen is not an active form. We need to convert it to become an active form. And that conversion, we talk about several conversion. We talk about the RAS, RAAS, renin-angiotensin pathway, RAS pathway, and then in that pathway, we talk about many enzyme to do the conversion. Here is the same thing. We need to have something to convert the fibrinogen to fibrin. However, this conversion is much more complicated than anything before, and probably the most complicated thing I ever read in the entire physiology. So let me show you this one here. So this is the all right, so I just changed the title a little bit. That make more sense now. So this process to convert the fibrinogen to fibrin is not just one, it's a cascade. So you can see that it triggered from the injured vessel, such as collagen exposure. There is several terminology that people talk about tissue factors, etc. But we are not going to talk about those. And it will go through a lot of coagulation, activation of several coagulation factors. So and that's not. And so you can see that these activation take a lot of a cascade. One act, the other act, activate the other, activate the other. In the end, the purpose is to convert fibrinogen to fibrin. So this a lot of check and balance. You got to have everyone to say yes, 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 yes. You need to have a lot of approval, and so only you collect all this approval, you can make them. So that's like a protection in a way that so it's not so easy to get blood clot. All right, so that's that. And so since this is kind of several cascade, factor, even though this one is already simplified version, I will only ask you the final steps. So basically that what converts the fibrinogen to fibrin, you need to know that thrombin. And the thrombin is converted from prothrombin. And the prothrombin converting prothrombin to thrombin is achieved by something nice, prothrombinase. So that this will be the thing that I will the final core action of this process. Prothrombin to thrombin, even though there are several other steps before, but these are the things that I will quiz about basically. After all, we activate prothrombinase. So just the reason I choose this near, right, is the core of this coagulation cascade. Is also something that you will commonly see that for the enzyme. Yeah, so it will be something nice. Prothrombinase, it acts on prothrombin, so it converts prothrombin to thrombin. Then thrombin will convert the fibrinogen to fibrin. So that's that. All right, so this is summary that we talk about. Several things will trigger these coagulation factor. Platelets is one of those. Activated platelets will also play a role there. Several coagulation factor, and also the basically the injured tissue will also involved. And so in the end, it will activate, it will convert the prothrombin to thrombin, activate the prothrombinase to convert prothrombin to thrombin. And then thrombin will then convert the fibrinogen to fibrin. And then we have the fibrin polymers, a mesh-like structure to enhance the blood clot. I guess that's it. I guess that's everything we need to know. Yeah. Yeah. So once we form it, right? Once we form this clot, the next thing is to dissolve it. Okay. You cannot just keep it there like forever, because it will be, once it's unstable, it's gonna cause the thrombosis in other part of your circulation. So we don't want it. We want to resolve it. There is a balance. There is optimal time, and so we're not. It's basically that these two forces usually just kind of like it's like a top tension, like the factor to resolve it is always there. You just need to give it time. It will resolve it, resolve this coagulation. All right. So once we form this clot, eventually we need to resolve it. So this is one we are going to talk about. We will need to resolve this clot. And to resolve this clot, we need something to cut this fibrin network. The thing to cut it is called plasmin. So where do I picture here? So here is plasmin. Plasmin will cut the fibrin network. And so when we cut it, you produce the fibrin degradation products, FDP. So you cut it, so it will get dissolved. So you don't have that clot, that coagulation anymore. However, plasmin doesn't present already in the blood. It's an active form, but what we have in the blood is an inactive form, which is plasminogen. So you need to convert plasminogen to plasmin. And what can convert that? Plasminogen activator. So PA is plasminogen activator. T refers to tissue plasminogen activator. This one will convert plasminogen to plasmin. and then you have the plasmin to dissolve the fibrin network. So that's that. So yeah, so that's that. Plasminogen activator or tissue plasminogen activator can is secreted by the endothelium. Basically, endothelium heal the wound, and so this healed endothelium will produce that signal, plasminogen activator. This one will convert the plasminogen to plasmin. Then plasmin will say, oh, we already healed it. Then you guys don't need to get stuck with us anymore. You can just get degraded. So plasmin will cut them back, and so you will make it smooth again. These plasminogen activator is also clinically relevant. It happens to patient with a stroke. So if the patient has stroke, all of a sudden facial drooping, motion disabled, and so when in that situation, you send the patient quickly to the hospital, do a very quick examination, then doctor can inject the TPA, tissue plasminogen activator, injecting it. It will circulating, it will activate the plasminogen, it will activate the plasmin, and then it will cut all this clot. So even there's a clot there, they will get dissolved, and then you can have the blood circulating again. So that is a very very life-saving way. So ischemic stroke is detrimental, but fortunately we do have a very effective treatment to treat it. You need to know that another type of stroke is called hemorrhagic stroke. So hemorrhage. If there's leakage, it will be difficult to treat. But if there is a clot just stuck on the brain, in the blood vessel in the brain, you can resolve it by injecting a TPA. But you need to do it early enough. If it's too late, you already caused damage to the brain, then resolve it is not going to do any help. So that's that. Quick question could be that which of following is the chemicals that can treat stroke, for example, acute stroke. It got to be acute, so acute stroke. So that's that. All right, so that's everything. Let me know if you have any question. Okay, so we finished the blood. The next one will be, white blood cells. We have not really talked about the white blood cell. We talk a little bit, but we will talk white blood cells combined with the immune system altogether, talking about body immune system, body defense in the next lecture. All right, so that's that. See you next time.